So the HT-CLI study is aimed at using cells that are capable of growing new blood vessels to treat no-option patients with critical limb ischemia. These are patients who are not amenable to angioplasty and stenting or bypass surgery that would otherwise lose their leg, and we know that up to a third of patients are susceptible to an amputation after they develop critical limb ischemia. So this new technology is aimed at delivering the cells that are able to grow new blood vessels in the appropriate region of the leg for those type of patients. So there have been several attempts previously over the years to try and use cells to regrow blood vessels for patients whose blood vessels have occluded because of atherosclerotic disease, but these have been unsuccessful. There are various reasons for this, one of which is that we've not yet found the most potent cell type that's able to promote robust growth of new blood vessels in the leg. The injections have been mostly single injections that are fleeting, and these cells don't remain in the lower leg for long enough probably to induce meaningful blood vessel growth. And the trials have been quite small studies, and they've not been randomized and controlled efforts. And so I don't think we, to this day, know whether cells are capable of effecting meaningful, clinically meaningful, that is, blood vessel growth in these patients. So this new study is aimed at addressing all of those shortcomings of the previous work. The trial is based upon recruiting patients who have patent blood vessels to the knee. So we're only aiming to grow blood vessels from the knee downwards, and I think that's important because previous efforts have tried to revascularize the entire leg, and I think there's no hope of doing that with cells using current technologies. However, below the knee, we think that providing multiple injections to the leg may well be successful, and that remains to be seen from the trial. So currently the trial uses a filtration system to filter the total nucleated cell components from blood from these patients. So we withdraw a quantity of blood, we filter the cells off from that blood to concentrate it, and we deliver these in up to 22 injections divided at equal distances from the knee downwards towards the foot. And this is repeated on 3 occasions, 6 weeks apart. The idea being is that we deliver cells, they start to grow new blood vessels, and then we deliver another batch of cells to sustain that blood vessel growth. This is going to be studied in up to 350 patients in multiple sites around the world, with close follow-up, including wound imaging, for up to 1 year. So we have a 12-month follow-up period that will determine whether there is any reintervention on that leg and whether indeed the wounds have healed or whether the leg has needed amputation. So we're quite confident that because this is a large randomized controlled study delivering a sustained population of cells, that we're hoping to be able to definitively answer whether this type of approach is able to salvage the limb or not.